This is IAQ Radio, Indoor Air Quality Radio, the voice of the indoor air quality industry, with your hosts, Radio Joe Hughes and the Z-Man, Cliff Zlotnick. And now, Radio Joe Hughes.
Good day and welcome to IAQ Radio Plus, episode 731. This week, we welcome back Dr. David Miller. We're going to talk about his chapters in the recent second edition of Bioaerosols Assessment and Control. The long-awaited update is loaded with current and updated information. Before we get started, let's thank our sponsors. They're the reason we can continue doing the show. And after the show, you can continue the discussion on afterthoughts.iaqradio.com. IAQ Radio Association sponsors are AIHA, the American Industrial Hygiene Association at AIHA.org. IICRC, the Institute for Inspection, Cleaning, and Restoration Certification at IICRC.org. The Restoration Industry Association, RIA, at restorationindustry.org. The Environmental Information Association, EIA, at EIA-USA.org. Actionable Insights at getinsights.org. IAQ Radio industry sponsors are Particles Plus at particlesplus.com. BioPlanet at byoplanet.com.
And now you can win a cool prize. It's time for the IAQ Radio Trivia question. Be the first to correctly answer. Simply email your answer to czlotnick at cs.com. Or if listening live, just text your answer from your computer. And now, here's the Z-Man.
Hello, everyone. I'm happy to report that Donald Weeks in Ottawa, Ontario, Canada, was first to identify a polymath as an individual whose knowledge spans a substantial number of subjects and uses complex bodies of knowledge to solve specific problems. Here's today's IAQ Radio Trivia question. Name the heat-stable bacterial poison responsible for the pathophysiological consequences of certain infectious diseases that Richard Pfeiffer intellectually and experimentally conceived. Back to you, Joe.
All right, today we've got Professor J. David Miller. He has focused his research on mycotoxins in damp buildings and health for the last 40 years, first at Agriculture Canada and then at Carleton University in Ottawa. He has published over 370 papers on fungi and fungal toxins and has co-written 10 books on the public health aspects of exposure to fungi and fungal toxins and has several patents. Welcome back, Dr. Miller. Great to see you again.
No, it's been many times now. So.
Well, we've been waiting for this show now for a while because we want to talk about the second edition of Bioaerosols Assessment and Control. And we had a show 2 weeks ago, I believe it was, where we interviewed a few of the authors, and I wanted to get you on to talk about your chapters in this book. It's a fascinating read. I've enjoyed some of it's kind of a trip down memory lane because there's stuff in there from the Green Book, and it seems like maybe there's even some carryover from the first edition. What are your overall thoughts on the process in the book?
Well, firstly, I'll say the obvious. It took way too long, and it's not Sherry, not Jack's fault. They worked hard. They nagged. They tried their best to get it done in a more reasonable time. But it is done, and that's good. I actually spent a huge amount of time on the 3 chapters I wrote. And also on doing quite a bit of reviewing and helping to find reviewers. So I think it's something everybody can be proud of. And I guess the last thing, Joe, is I actually was the lead peer reviewer on the 1998 manuscript, which I still have. I remember offering many suggestions for things that should be fixed. I'll say I was only partially successful, but I too have a copy of it on the bookcase behind me, and it has been helpful for providing some direction for AIHA and other organizations to know that their advice was on mold and dampness and whatnot was being advanced. I guess the other thing I'll mention is that Sherry mentioned in your interview with she and Jack that who would have thought that there'd be a chapter on mold on marijuana production facilities back in 1998, right? So
yeah,
anyway,
yeah, things have changed quite a bit over the last 25 years. Well,
they sure have.
let's focus on that last 25 years. Chapter 2, the basis for health concerns, was one of your chapters. Tell our audience, what are the most important points of what we've learned about allergic disease, or AKA building related illness, in the last 25 years?
Well, I'll just separate out allergic disease. I think that a couple of things. One of them is because we, in North America, tightened our buildings up, added carpets, and shifted to paper-faced gypsum wallboard in our working lives, right? Is that provided a home for dust mites, for example. The first time they were detected in North America was in 1969. Now there's hardly any building in North America that doesn't have a decent burden of dust mite allergy. As with always things in medicine, frankly, it did take a long time from recognition to widespread acceptance that they caused allergic disease. A second thing is I would actually say on mold. So as I often say, when the first epidemiology studies were done in North America by Health Canada and by the Harvard School of Public Health, that seemed to indicate that living or working in a moldy building increased your risks for upper respiratory disease and allergy to everything, not just mold, that was considered so ludicrous it couldn't be true. And a big change was the investments made to understand if this was real or if there was something else that we were missing. So I doubt if there's any plausible organization or physician in the world now who would say that we're not sure that mold and dampness causes allergic and non-allergic effects. We're sure, and we know why. So that's a really big change in the last quarter of a century.
In my experience, indoor environmental consultants commonly forget about dust mites, cockroaches, and other common allergens when looking at buildings versus homes. Sometimes when they're in homes, they'll look at these, but rarely in buildings. Can these common household issues also cause problems in larger buildings?
Yes, they can, and it's something you always need to keep in the back of your mind if you're dealing with unresolved complaints. And this comes up in the chapter I drafted about exposure in workplaces that deal with laboratory animals. So if you think about peanut allergy, everybody knows there are people that are what are called exquisitely allergic to peanut allergy. In other words, if they get exposed and they don't have epinephrine nearby, they could die. So it works out that there's a cohort of very sensitive people in the population. And then if you bring you're coming in and your clothes are at home, they're carried a lot on clothes into a workplace, so it can happen. And I recall a case that the late Phil Morey had been drafted to try to understand. That was an office building, and after everybody tried everything, and then finally Phil said maybe we should measure dust mites because it turned out there were a few people in that particular office who were allergic to house dust mites. And of course they were there. And if the building isn't maintained properly, then the food for dust mites, which is skin scales, accumulate there as well. So it's a confluence of the risk of finding somebody that's quite allergic to house dust mites or cockroaches. Um, um, a workplace where you have a sensitive person, as I said, and then whether they grow there or they're dragged in there or not, it doesn't really matter. And the clearest example of that is that people working in animal production facilities or even pet stores, it turns out, drag their allergens home, and you get some signal of increased sensitivity in their kids, for example. So that can happen. It's a thing to keep in your mind if there's unresolved complaints after a very long time.
The basis for health concerns chapter it wasn't just on, mold, obviously. There were a lot of different health concerns. I found the discussion of relative humidity, temperature, and viruses very interesting. It seems that RH is virus type dependent, I guess, or the effect of RH is somewhat dependent on the type of virus. Can you talk to our audience a little bit about
it? So this is important, and I did, in fact, file in the breathtakingly short public comment period for 241 a fairly long comment about this absence. So it turns out, as a generalization, that survival of the viruses that get into the air, including the ones that were thought to be or that were examined for biological warfare purposes, is that there's a curve like this. So survival turns out to be lowest in general at 40% or so. But if the humidity is higher or lower, the virus can survive in air. And that is a true generalization. And so in the papers I cite, these are very detailed studies that support that. And it was a lot for military reasons this work was done. We also see it epidemiologically and seasonal distribution of viral infectious disease, respiratory diseases. So it matters quite a bit in a built environment that the humidity be maintained in the 40% range to decrease the survival of viruses. And there's absolutely no doubt about it. There is a second piece, and that is our susceptibility changed if the humidity is higher or lower? And the answer is, at least based on relevant animal models, that the answer can be yes. And the curve isn't quite as sharp as that, but it still tends to be that the range that we would like for mold and dampness, for example, is a good range to keep. But if you go too high or too low, then that's a problem.
Interesting. Cliff, did you have a
Yeah, no, I have a follow-up. Yeah, it seems that I guess over the past couple of years, I think there's been maybe some reconsideration of older research work and so on and so forth regarding relative humidity during the winter time. A lot of times, flu and some of these viral things are worse during the winter, and particularly where we're at in the Northeast. It's pretty dry in the wintertime, particularly with some of our heat sources, if they're using hot water heat and so on and so forth. So it's actually been recommended to increase the amount of relative humidity, during the winter. And supposedly a benefit to doing that was less risk of respiratory disease, which seems to be maybe not entirely in parallel with your findings.
Yeah, so firstly, it's not my finding, although I was involved in the research for reasons that we'll just leave alone for now. But a couple of things there. And remember that I worked on the Quad AI committee for environmental allergens. And there are papers on this, Cliff, but basically the advice to increase humidity when you're sick, it turns out to be an old wives' tale. So this was the common advice, certainly in our age, like your kid's sick or you're sick and your mother brings a big humidifier into your room, and that was the advice. But there are studies that happened um, happened in in my time where this advice was looked at really critically, and it doesn't stand up. So, I mean, I apologize for this, but Queen Victoria got pregnant quite a few times, and towards the end of her whatever seventh kid, she sort of asked her physicians, who would have been the best physicians in England, how to avoid it other than the obvious, and they told her to have sex in the middle of the month. So that's a parable about lots of things get said that just are actually wrong. It's just that they get sick, right? So for both susceptibility and for a humidity that's comfortable for mold and dampness and not necessarily clogging the dust mite population up, that's the right humidity.
All right, we're gonna get to fungal metabolites in a little bit. I see I've got a good question on that, but I want to first finish this chapter on health concerns.
Right.
Let's talk a little bit about humidifier fever, endotoxin, and bacteria in indoor environments. First, what are the sources of exposure to endotoxin, and is all endotoxin the same?
So in a house, so I'll start by emphasizing this is important. It's been known since the middle of the 90s that if there's an amount of allergen, cat, dog, cockroach, dust mite, and there's more versus less endotoxin in the dust too, you will have more symptoms. It's been known since the middle of the 90s for sure that if you're a kid who might have the predisposed allergy, atopic, that if there's more versus less endotoxin, then the amount of allergen to elicit response to set you on the journey of being allergic. So this really matters in a population health context. And it's one of the reasons why in our housing and health research studies, endotoxin is a very important analyte. And that there's no ambiguity about it. It's been known for decades. The sources of it, it depends where you live. In a more rural area where there's farms around, there's endotoxin in dirt, so it gets in the air, it gets in your house. Pets drag it in. It generally comes from either outdoor air, from the dirt, or from tracking it in, and pets. It doesn't proliferate in the building, it just comes in from outside, which is why one wants to, if you have carpets, to really be careful to reduce the burdens of fine dust, which is where it hangs out.
So in the book, there's now a separate chapter on fungal secondary metabolites.
That's right.
And I'm trying to understand, is endotoxin a secondary metabolite from bacteria?
Well, it's from gram-negative bacteria. So in your house, or there's not many people in my building at Carleton right now because it's between terms, we drag in lots of bacteria. So that gets, a lot is known about that. People used to think that was a really bad thing. It's nowhere near like as bad as endotoxin. The thing about endotoxin chemistry, though, that is super important. So endotoxin exposure in workplaces, for example, a sewage treatment plant, a pig, confinement, a barn that's full of cows, a compost, composting commercial operation, the endotoxins that are present there chemically are different And the potency of those different mixtures varies by 2 orders of magnitude. So it really matters the distribution of bacteria that are gram-negative that make endotoxins, where they come from. In other words, what might make sense for a big production facility versus a cow production facility versus a compost facility versus residential accommodation. They're entirely different chemistry. So we use one word, but there's I think it's around 250 distinct chemistries that are a function of the particular gram-negative bacterial species involved.
Should there be a TLV for endotoxin?
Well, in my opinion, the answer is no. So for, the potency of a mixture that, for example, might be in a pig production facility, it could be quite a bit greater than what's in a home. So suppose somebody comes in and makes a measurement and says, oh my God, we're all going to die, but they're not measuring the same thing because the chemical test that we use, or the test that's used, which comes from the horseshoe crab blood, is not capable of distinguishing the fine chemistry, merely how much endotoxin is present.
Hey, Joe, if I may, I have a question.
Go ahead.
Years ago, when we used to do realistic training, we would, we would simulate sewage contamination. And
Okay.
what we were trying to do is have sources of endotoxin and bacteria in this room. And I told a friend of mine who's a microbiologist what I wanted to do, and she said, I don't think you should do it exactly the way that you're doing it, but there's a safer way to do it. And what her suggestion is that rather than using human fecal matter, she suggested that we use horse manure. And so what we would do is we would gather fecal matter from horses and we would mix that with the water. And apparently it was her opinion that either there were few or no known pathogens that would pass from this horse manure to humans. So I guess not all, I guess the point is, does horse fecal matter produce endotoxin? And if it does, is it less toxic than others? Or?
So let's separate. I mean, I cautiously agree with not using human.
Right.
Because it has human pathogens in it.
Right.
Including things like verotoxin producing E. coli, for example.
Right.
Although that said, the epidemiology studies that the CDC and the NIH does after a major flood disaster, we generally don't see many people getting sick from the sewage in their houses, but they get sick from poor quality drinking water because it's not available. So let's remember that the humans, the real risk there is that there could be a human pathogen, and although the risk would be low, it's not wise. So horses generally aren't carrying human pathogens, although they could carry the verotoxin producing E. coli. Cows certainly do. And then for endotoxins, I wouldn't look in shit for endotoxins. So the endotoxin are this group, so-called gram-negative bacteria. They tend to be soil bacteria. They're environmental bacteria, and the important thing is that they don't grow at 37 degrees. So their lifestyle and their habits are really, really different. So endotoxins are, for the bacteria, are part of the cell wall or cell membrane. These are really old organisms. They're not necessarily there for the same reason, if you like, than fungal toxins are, which are to tell the competition to go away.
Right.
It's like the fungal glucan story. For the fungus, it's just something to help build the cell wall. For us, it's a world of hurt that mold glucan is.
Our restoration, I was reading in your chapter, our restoration audience will be happy to see that effective cleaning of porous surfaces
Right.
and appropriate maintenance of humidifiers are the keys to controlling endotoxin. But my question is, what is effective cleaning of porous surfaces?
Yeah. Well, it's cited, but I asked that question after a study done in the Seattle, in the county where Seattle is located, where they gave people whose kids from poorer backgrounds kept coming to emerge for asthma attacks. So they gave them HEPA vacuums, and then they had two arms. One where they taught the people how to use them properly, and then let's just give them the equipment. And both of them, it actually helped reduce the the need for their kids to go to emerge quite so often. So I wanted to know what that means. So we recruited some families and then offered professional cleaning um, and this is discussed in one of the AIHA documents from a commercial perspective. And we hired professional cleaners, and the volunteers were asked for permission to have professional cleaners clean their house every week for 6 weeks. There was no difficulty getting volunteers for that. And they did that with the best, basically commercial, residential HEPA equipped vacuum you could buy. It cost about $1,000. And these cleaners, they were totally into it because it's their job, right? And I had an engineer with a stopwatch to make sure every week they covered the areas in the house exactly the same way. And it actually took 4 to 6 cleanings by a professional cleaning to reduce it to as low as could be reasonably achieved. And just to drift back to endotoxin briefly, we chose homes that didn't have pets. And 1 week, the data, when I got it back, the endotoxin signal had gone way up. So I told the team, go find out what happened. It turned out that the family took the dog of the daughter who was away on vacation. And so we could even detect that the dog appeared in the one of the week's sampling. So that people overall underestimate in a house with a carpet that's older, that's where your exposure to outdoor air pollution comes mainly today in North America, because unlike when you and I were young, the air pollution has gone down every year since 1978 in North America. It's where they get exposed exclusively to endotoxin, to dust mites, to other allergens. So reducing that is, so we have looked, and I don't know what the advice from cleaning organizations is now, but the hope was that after they clean a porous surface, that they take a sample so they could check at least to see what the burden was. I think there's a number somewhere, and that number came from the Seattle King County study of human health that I mentioned.
Interesting. Cliff, you had another question on this chapter, I think.
Go ahead. I did. If I'm not mistaken, one of the things that we've done in fire restoration for many years is in conjunction with exhaust ventilation, we would spray aqueous and alcohol solutions, for example, ethanol or isopropanol, onto surfaces while we had a significant exhaust ventilation. The purpose of this was to desorb odors and VOCs, that might have been trapped into these materials. And when I saw your chapter in the book, it mentioned that the use of ethanol to detoxify and denature various toxins. And I was just wondering whether you would recommend, this application of alcohol and aqueous solutions as an addition to fungal remediation for sensitive clients.
Well, I think the short answer is no, because firstly, I would use the WHO language, it's undesirable and not necessary. I mean, the guidance since New York in 1992, when Dr. Mori and me and union guys from the New York City and the New York Department of Health met, remove the damaged material and clean it thoroughly, and document that you have done that. The problem with adding chemicals, and this is discussed in the AIHA thing about floods or disasters, it should be there somewhere, is that if you think about bleach and you added it to your water with horseshit, how much bleach would you have to add, the active, chlorine residual, to actually kill everything, given there's so much organic material there?
No, it wouldn't do it, right?
It wouldn't do it. So,
wouldn't
so
do
what
it.
you always have to think of, unless it's a smooth surface like a countertop or, that you're just wiping off, then basically every bit of the active in your biocide, whatever it is, will be killed off by the dirt, and it just won't do what you imagine. And the danger of that really is that a false sense of security, rather than perhaps not necessarily causing harm.
Let's go to halftime here. We're going to stop and thank our sponsors. We'll be right back with the second half of our interview. Association sponsors are AIHA, Healthy Workplaces, A Healthier World, AIHA.org. The Environmental Information Association, EIA's multidisciplinary membership collects, generates, and disseminates information concerning environmental and occupational health hazards in the built environment at EIA-USA.org. The IICRC, a nonprofit standards development and certifying body for the cleaning and restoration industry, IICRC.org. The Restoration Industry Association, the oldest and largest nonprofit professional trade association dedicated to providing leadership and promoting best practices through advocacy, standards, and professional qualifications for the restoration industry at restorationindustry.org. Actionable insights, no more mistakes, no more missed line items in your Xactimate estimates at getinsights.org. Industry sponsors are Particles Plus, feature-rich particle counters and air quality instrumentation. Count on us, particlesplus.com. BioPlanet at byoplanet.com, improving human, animal, and environmental health with electrostatic spray technology and advanced chemistries at byoplanet.com. All right, we're back with Dr. David Miller. I gotta go into chapter 24, and first I want to kind of go over why you changed the title of this chapter from what was fungal toxins and BD glucans 1 through 3 in the old edition, and now it's called fungal metabolites. What was the reasoning behind that?
Well, firstly, I didn't write the 1998 version. Let me tell you for sure that I would have the Pearl Harbor file on this, so I'm not making this up, that I counseled against it. But that was the fashion in the time, not my fashion. Because, as you know, since we've talked about this over the years, I know a great deal about the fungal toxins on food. And there's a strict definition of that, and that is a compound known to cause health effects in human and animals. And this is in my chapter as well, that people who talk about mycotoxins, and even the people who measure allegedly toxins in urine of sick people, mycotoxins, they're measuring toxins that come from food. My discussion in my chapter is quite a comprehensive discussion of the metabolites that people talk about from fungi and what they mean from a health perspective. And I mentioned, I think, Joe, in a recent email, that recently I'd seen someone who wrote a report that claimed that the volatiles were super toxic, which is completely wrong. But it's the kind of thing that people, went about to say, and still do. So,
Well, let's go a little bit, because there's a good bit of information on microbial volatile organic compounds,
right.
MVOCs. What are the key points that our industry should understand about these MVOCs?
Right. So a key thing is that humans can smell mold vials at levels, concentrations that a machine on the planet can't. So we're very, very sensitive as a species to moldy odors. And that's a good thing, because eating moldy food is a bad idea. And the clinically and the quad-AI reports say all this, that and Mark Mendel and what we say in the Green Book is that mold odor is a very useful signal for supposing that there might well be a mold problem. The other thing, which I'm sure that Sherry Marsham and others just recently updated the odor threshold ACGIH book, I guess, and we can smell these chemicals from molds again at levels that are 2-3 orders of magnitude from where a TLV would be. So they're mostly straightforward organic. Like one of the most important volatiles from many fungi is ethanol. Okay, but it's the mixtures that gives the sense that there's something moldy here.
And as far as the so I guess I look at the MVCs now as sort of like CO2, in that we use CO2 as an indicator for lack of ventilation. MVOCs are an indicator of mold contamination. Is that
That's right,
accurate?
and that's an accurate epidemiological fact.
All right, I got a text question I want to hit you with here. Based on your pioneering work, mold response starting in the 90s emphasized the risk for mycotoxin-related illness and need for mold testing to resolve IAQ concerns. Reflecting back on what we've learned since that time, do you feel that occupants exposed to mold growth experience toxicity symptoms? And is mold testing still needed to assess mold exposure and clear remediation?
Yeah, so to add to the Green Books and the original 1996 field guide, testing is this has been in everything I've written since my first talk at ASHRAE in 1992. No one should test anything until they do a proper informed inspection, meaning someone qualified in that type of building. And I can't account for what has happened in the marketplace since. The second thing is that testing is not always necessary, but there are reasons in the Health Canada guidance. If you can see the problem, then why would you test? The short answer would be perhaps not. But suppose somebody's suing somebody, then sometimes tests are needed for other reasons. And this is in the guidance documents. But you start with an inspection, you start with an informed inspection systematically. If a lot of problems in big buildings, like I can think of a problem where the problem in the building, which made a lot of people complain and have to leave work for illness, was that the interior of the duct was full of active mold growth. You couldn't see it. The only way we detected it was to do some sampling and testing. So sampling and testing is an essential part of the repertoire of a mold. What the official guidance, at least in Canada, is that homeowners should not be testing for mold, because normally it's not necessary. Again, a lesson until there's legal lawyers involved or something like that.
Yeah, I think that's a problem we run into a lot, is especially with school districts. They want some numbers, they want some lab results, and that makes it difficult for those of us in the field. We're going to get into I got a couple more texts, but before we do, I want to add, what is community?
Yeah. So if you are working or living in a moldy house, and it speaks a little bit to one of Cliff's questions, and it's bad, so you need to leave. And then they clean, they do the best they can, but there's some residual volatiles, for example, like MVOCs that are in the fabric of the building. Once I saw a case where they took all the documents and paper and equipment out, remediated, but they brought all that back. So some occupants could sense by smell, if you like, that the threat was still there. And there is a real phenomenon. It's important for, arguments about whether my pig farm is causing you problems or not, if you can smell the odor, is that if your body is exposed to an odor repeatedly while you're actually being threatened, by, say, mold particles, then the body thinks that it's still being threatened when it's not. And for that reason, I would encounter situations when I was in the government in Canada that you could find 1 in 100 people that couldn't go back to that space because there were, the association of a small amount of maybe even perfectly ordinary volatile will. So it's a real phenomenon. It's actually mentioned in the first edition in Harriet Amon's chapter. And just always keep it in the back of your mind that it could be this rare thing is that, and not a person being crazy or unreasonable.
All right, let's get back into these fungal metabolites. What are the most important fungal metabolites when it comes to health effects?
Yeah, so the answer is fungal glucan. And that matters even for Ed's talk. We all know, I hope, that we're from the spring until snow flies in our neck of the woods. There's all kinds of fungal spores in the air. The spores that live on the surfaces of leaves, or the jargon is phyloplane fungi. Another big source of fungi are mushroom spores. So about 5-6% more of the American population are allergic to the fungi in outdoor air. Like you get hay fever, if you like, right? It turns out that the molds that are dominant in damp building materials, they're from a different taxonomic classification than the dominant phyloplane fungi and, of course, mushrooms. And they have, as their structural, one of their structural components is this compound beta-1,3-D glucan. And it turns out that, like an appendix, we have a receptor in our lung that in medicine is called the dictin receptor. So that when we breathe fragments of mold, meaning the fungi from damp buildings, spores, it turns that receptor on. And that sets in motion a very unattractive immunological cascade. And so that's a reason why, again, in research grade studies, we measure true fungal glucan, mold glucan, which is chemically different than found in mushrooms, than found in plants, than found in the dominant fungi, the phyloplane fungi. So that really matters in terms of health effects. It's turning on the immune system in an unattractive way, among other things. And there is, again, no
And I also want to ask about the difference between spores and spore fragments. And I believe in the book it says that the spore fragments are much more prominent. You know, there are a lot more of them, I guess is the terminology
Well,
I'll use.
and importantly, they get deeper in your lungs.
Okay. Okay.
Because remember, a spore is relatively big, and it gets stopped either in your nose or in the upper part of your lungs, but the particles will go deep, and huge amount is known about this because of mainly EPA studies on outdoor air pollution, and they concentrate in regions in your lungs and that's where they're causing a problem. So there's two variables. One of them is in a house without a mold problem, most of the spores would be intact, and almost all of them would be from outdoors, as well as you're tracking them in when you walk in from outdoors. That's where the Aspergillus, penicillium, whatever live. When it grows, then and dries, then the mycelia and the spores break apart, and that's what forms the fragments. So it turns out then that the majority of exposure to glucan as a proxy actually is in the respirable range in a house. So it's getting deeper in your lungs and causing chaos in your lung biology.
Cliff, I want to make sure you get a chance to jump in if you had a question.
Not about that, Joe. I didn't.
All right. Let's get back to the mycotoxin topic for just a moment. I think in the book, and I think you would agree that at high levels, they can be a health issue, but there appears to be an overemphasis today in the industry. And I'm wondering what your opinion is of, I got a question from one of our audience here. What is your opinion of ERMI, for instance, the Environmental Relative Moldiness Index?
Yeah, well, let's answer your question first.
Okay.
So again, I've staked out the territory that since 1953, the definition of mycotoxins is a compound known to cause human or health problems. Okay, and those in North America are well, in the world, are exclusively from food. If everybody in this audience is exposed to if they eat wheat, is exposed to the dominant fungal toxin that occurs on wheat, because we can't tell God to stop making it, so we manage it very well. In developing countries, people die, not tomorrow, today, kind of thing. So that's what we're exposed to. So for example, when I see damp building reports where people are claiming that they got exposure to the fungal toxin, ochratoxin, in the building, no, they got it from eating wheat that has, in North America, rather little of it. In Europe, it'd be a lot more. Notice that in anything I would have written, and there's even structural pictures of the structures in the book, the compounds that are made from by damp building fungi, about which I have done more than anyone in the world. These are low, I would call them low molecular weight compounds. And in my chapter and in other writings, after spending 10, 15, $20 million, they can play a small role in the health effects, but it's really the glucan that drives it. They're not very toxic, and they're not, they have some effect, but and so I give the specific example of Stachybotrys, which people talk about. The toxins from Stachybotrys are in fact quite toxic. The toxin workplace exposure to Stachybotrys in the olden days on farms and stables, but also there were cases from, I don't know, greenhouse facilities in Europe and agriculture in Africa. They are very dangerous, and you can be affected seriously by them. But interestingly, at levels that matter for, that could occur in a building, except during remediation. So there's no doubt about that too, for the same reason for Stachybotrys. But some good work was done on the biology of the effect of molds, and they used intact Stachybotrys spores, in other words, that had the toxins, and then they extracted them. So it was only basically the glucan left, and the glucan explained almost all the effect because it's so potent.
Why? Well, never mind. I'm not going to ask that. All right.
Oh, it's okay. Go ahead.
Well,
Be
then
brave.
why the focus on mycotoxins in industry and in laboratories and so forth, as opposed to the fungal glucans? I don't see anybody advertising that they do fungal glucan analysis.
Yeah, well, I can't apologize for that. This is what's in what the literature says. This is what I've been involved in saying for a very long time. This is what the American Quad AI panel report said. These are the things that matter. And the other danger is that people say invoke a toxin that exclusively comes from food as evidence that somebody is getting it from a building, which is just not true.
All right, I got a text here. Some mycotoxins can act synergistically with other microbial metabolites and even sensitize mice to protein allergens. How can these synergies impact exposure?
Well, I can't defend that statement. I helped to write the rules internationally about this, and synergy is a word that people toss around, but there are very few true examples of it. And again, if I just contrast the glucan, remember what I've shared, that here's a chemical, it's broken into small pieces, gets in your lung, and it like the affinity for beta, the mold glucan in that receptor is so freaking high that it'll turn it on even with a little bit. I mean, the other thing, which I've already said, is that it's been known reliably for a long time that if you live or work in a moldy building for a long time, that you're more likely to be allergic to everything. But that's again a function of what's happening biologically in terms of messing with this critical receptor, which we don't need anymore. But it's like an appendix. It's just we have for the last couple 100,000 years, or millions of years actually.
All right. What about people who are out working in the field? Do you recommend they learn how to do this fungal glucan testing and do it more often? Is it something that may be of use?
Yeah, I think the answer is yes. Yeah, I think in the Green Book there's a little discussion of it, and there are, I think in my chapter, yes, from the 280 references or whatever it is, there are actual human exposure studies, meaning chamber studies, small ones. One of the people who did those studies said that someday we'll be able to figure out a beware level. It's a very sensitive way of knowing whether there's exposure if you've cleaned the building well enough. All of the housing and health studies we do, we measure it properly. There's a couple of ways to do it. Some are better than others. And I do think someday that we'll add that method to our repertoire of measurements.
And what is the best method for that?
Yeah, well,
That might be a whole show.
It's a long, complicated, but it's actually, so it's based on the same test that we use for endotoxin, because the limulus, the horseshoe crab, its primitive immune system for fungal invasions involved the dectin, something close to the dectin receptor. And so basically what the test does is, or at least the test that works, is it's measuring, destroying the endotoxin in your sample, and then what's left, the assay is measuring the medectin.
All right, we're going to have to go to the roundup, but Cliff, I want to make sure you get a chance to jump in. I like this last question too. Let me get this in. Has epidemiology established that occupants who are not atopic,
Yes,
experience mold-related
100%
symptoms?
yes.
100%. Very
And
good.
non-atopic asthma, this was another conundrum with the two big original epidemiology studies from Harvard and Health Canada. That made no sense. But we started to see a rise in what's called non-atopic asthma. And the really good NIOSH studies of the Connecticut office building brilliantly portray what I just described, that if those exposed who were not atopic had the increased risk of allergy to other high exposure allergens they were exposed to, including outdoor air allergens.
Fascinating. All right, let's go to the roundup, Grayson. All right, Cliff, let me turn it over to you. I want to make sure you get a chance to ask a question, and then I want to give David a chance to talk about Chapter 20, miscellaneous allergens and irritants.
Okay. I think that this question, it's pretty basic, but I think it will be helpful to everyone. And David, what's the difference between an antigen and an allergen?
Yeah, so an allergen is a compound that is a protein, almost all. There are few exceptions that are that raises IgE antibodies. So it's a technical thing, whereas an antigen is on the journey to becoming an allergen. So in other words, it hasn't recruited all the machinery necessary to give you the linkage to unattractive allergic responses, isn't quite there, but it's raised, like, if you like, an alert in your body, and with sufficient exposure, then and it will light up, and then you'll become allergic to it. So it's something. So in other words, all allergens have to be antigens, but some antigens don't end up proving to be allergens.
Yeah.
We may run over about 5 minutes. Is that okay with you?
Sure.
Great. Chapter 26, miscellaneous allergens and irritants. I got a chance to look at that last night, and I found it very interesting, actually, especially the section on outdoor pollen and allergens in the outdoor air. But I wanted to give you a chance to tell our audience about some of the key points from that chapter and why it was added.
Well, I think about marijuana production, commercial marijuana production, it was a workplace that didn't exist in 1998. And as cited in the chapter, there have been, a number of health hazard evaluations by states that legalize marijuana, and in Canada where it's legal to produce it commercially, and thus a whole workforce that needs protection, exists now that didn't exist in 1998. So it's a pretty comprehensive analysis of what's known, what you should look for, and to protect that worker population. And the other thing is about animal production facilities. They're obviously in universities, labs that do toxicology testing for drug and chemical companies. So it's a fairly big place as well, and so that can elicit problems, and so there's material to help industrial hygienists manage that better.
Now, plant pollen, as I clear up my runny nose here because I'm terribly allergic to it. I noticed in there you had a pretty good detailed outline of how that has actually gotten to be a bigger problem over the years.
Well, and it's something that's been discussed at the Quad AI since 20 years, and that is that climate is, I'm from the eastern in New Brunswick above Maine. And I'm ragweed, which is the most important pollen. It's the one in the late summer and fall in North America in terms of, and I think it's the biggest over-the-counter drug spend in the United States and Canada. That literally didn't exist there. In the 1950s, Ontario used to advertise to people living in New York that there was no ragweed up here, but it's not true now. And so one thing is that the plants that are cause allergy move north. A thing that perhaps is less important, but a little bit important, is that irritatingly, ragweed makes more pollen per kilogram plant when the CO2 is higher versus lower. And when the NOAA started to measure CO2, let's remember it was 178 ppm, and it's, you know, quite a bit bigger now. So we have that climate moving allergenic plants around has caused new populations to be affected by hay fever and worse. Yeah.
Now, is pollen similar to mold spores in that there are also smaller particles? That...
Good question. They're big, but it turns out when it rains, they blow apart because the hydroscopic, they're dry, and they people taken films of this. They just literally explode into a bazillion tiny pieces, and that's why one of the reasons why you often have more symptoms after thunderstorms.
Now, my final question, Cliff, do you have any you want to get in before?
No. Are there any text ones that we need to?
I think we covered most of that.
Okay.
If you take a look, though, while I'm asking this one, a second human case of bird flu in America is raising some alarm. How close is the H5N1 outbreak to becoming our next pandemic?
Well, I think there are those who argue that we should be working on a vaccine. I saw a report from a scientist in the University of Arizona that we should be working on a vaccine now. And that the other thing that, because he's been doing sequencing like the early days of COVID, that it was here, well, it was in the United States sooner than we thought. I think if, you know, humans, you know, the history of those who don't learn from history are doomed to repeat it. But all of these, Cliff and you and I, are old enough to know that we, as a species, don't really think very hard after the last disaster and just proceed on our way as if it can't happen again. But I think that lots of lessons were learned, and I think that, I guess, I hope that our officials are on the ball and planning for when and if we need a vaccine. The great thing is that is you're drinking pasteurized milk, there is no exposure whatsoever, and the risk at the present time is basically workers who work in dairy production facilities or otherwise handle cows, and so they'll need some special attention.
And I guess the concern is also the mutation of that virus.
Yes. I mean, all of us, at least you, hopefully get flu shots once a year at our age, and you know that they have to change it every year because it changes. And they try to match the strain. They don't always succeed, but they do their best. And that's what viruses do. They're out to get us. and you know, H1N1, like all our other viruses, came from China, where you have animal production facilities in very crowded environments. And that's a recipe for what we've had since Spanish flu.
Dr. David Miller, I really appreciate every time you join us. I learned something new. Before we go, is there anything we missed that you'd like to add?
Well, I hope I've tried to, I mean, you came up with a long list of questions that would take usually quite a bit longer. But I think let's just focus on the fungal metabolites. The only one that really matters on mold is beta-glucan. Let's think about mycotoxins, and AIHA is redrafting its mold, white paper or whatever it's called in AIHA terms. The things that people measure or talk about as mycotoxins, they're from food. They just don't occur in buildings. And as noted in my chapter, the low molecular weight compounds, including the actual toxins, because they do cause human health problems from Stachybotrys exposure, that is not a major part of what we see. It's the glucan that causes that.
All right. I want to thank our guest, Dr. David Miller, my co-host, the Z-Man, Cliff Zlotnick, Grayson, Gone Fish, and Fisher at the controls. Most importantly, our loyal audience. We had some heavy hitters on today. Nice group of people listening in. Thank you all for joining us. Next week, we've got, let me think, Seth Harrison from Actionable Insights. We're going to talk about some current events in the restoration industry. Please come back and join us next Friday at noon for the next episode of IAQ Radio Plus.
For IAQ Radio, I'm Spike Reel saying thanks for listening.